My name's Alan. I'm based at the University of Bristol in the UK. And I'm here to talk to you about the Regenerate PD study. So thank you so much for the invitation to Helen and Cure Parkinson's and the Andel Institute. Thank you very much for the audience for being there. I'm really excited to be talking to you about the Regenerate PD study. And the reason I'm excited is because I think it has the real potential to alter the management of Parkinson's. And I'd like to ask for your help. I mean, you're obviously an extremely committed group of people to helping Parkinson's. And if you can, through your contacts, spread out the word of the Regenerate PD study, that may help our recruitment. And if we can recruit quicker, we can get to the end quicker and potentially get this therapy to clinic. This is a document that was put together by people with Parkinson's and caregivers, charities, and academics. And it's a terrific piece of work where there was real thinking about what do people need to know if they're going into consent visits for clinical trials. What are the key questions to ask? And you can get this document from Helen if you wish to. Now, the first key question is, why are you bothering? Why did your mother give birth to you? What was the point of turning up to have this trial? And what this trial is all about is neuroregeneration. So this is pretty exciting. This is regenerating damaged brain cells, damaged dopamine neurons, which, if they are sprouted and grow, could potentially push the disease back to before you had the condition, to have you better than over baseline and maintain at that condition state. And that's obviously different from symptomatic therapies like L-DOPA, where although they make your movement symptoms better, of course, year on year, people are declining. It would be terrific if we had a neuroprotective agent, and CP are very much helping develop some of those, but at best they're only likely to slow the condition in its progression. Good, but you're still going to be getting worse over time. And it may be the answer to Parkinson's is to have a combination of neuroregenerative and neuroprotective therapies taken by the same person. Now, in terms of the therapy I'm talking about, the treatment arm, which is in the virus, is GDNF, which is a trophic protein. So these are proteins, a bit like Baby Bio, which is something you can get in the UK at least, that you put on your tomatoes, and that helps nerve endings to sprout and grow and regenerate. People who found these growth factors are both shown, and they won the Nobel Prize for this. So hopefully that gives you encouragement that there is pedigree in the background to this type of approach. That said, to date, 5 double-blind trials, where double-blind trials are those studies where you have a portion of patients on the active treatment and a portion on placebo. 5 double-blind trials, although some signals of encouragement, have not yet shown clinical significance to take these forward. Now, for 4 of those, importantly, we may well have found the solution to why that happened, and that's to do with delivery. And to explain that, we have a PET scan, and you've got 2 panels there. Now, to answer the question the gentleman asked, how do you get this into the brain? You have to have a surgeon. You can't get it into the brain by intravenous therapy or orally. So you have a surgeon implant the therapy. And in this study, they've implanted GDNF into the yellow highlighted area in the left panel. And the green around that yellow area in the center of it shows that there are no dopamine nerve endings there. And as the GDNF is poured on continuously over a couple of years in this trial, you see it goes red in that yellow, which is dopamine nerve ending sprouting. But that target structure, the putamen, which is about 3 cubic centimeters, spreads up to where the top end of that red is. So the problem is you've got only a very small area of regeneration, and the drug didn't spread through the brain. So the development, therefore, which was designed by the chief scientific officer for this study, was something called convection enhanced delivery, where that approach would allow you to spread the drug through the target area. And we believe with a study in Bristol that I was involved in, we did do that. So in that study, patients had a port behind the ear. You can see the port behind the ear of one person there, a bit like the port in the Matrix. And every 4 weeks, they had a pump clipped on and via syringes, drug GDNF pushed into the brain down cannula, which is shown in the red area of the schematic. And on the right-hand side in the MRI, you've got gadolinium dye. And you can see that's filling, therefore, using this convection-enhanced delivery approach, filling the whole putamen structure. Now, that's unprecedented delivery. So via convection-enhanced delivery, we can get this drug into the brain. But does that, but is this the way to go forward? Because for this type of delivery, you have to go for every 4-week infusions, which once you get to clinic may not be satisfactory. And here's the proof that it did get through the brain. These PET scans were from our CED study, and what we're showing in the top panel is that the dopamine terminals have been lost, and it doesn't change between placebo at baseline and placebo at 38 weeks. But in the bottom panel, we have people on GDNF, and you can see between baseline and 38 weeks, that red spreading all the way through. So you're getting target tissue engagement, you're getting a biological response. Again, I hope this gives you confidence. But once again, the clinical outcomes were negative. Now, what we're looking at here is diary time. So on the y-axis, this is good quality on time. So what you can see, so this is when it's good quality on time. It means when you're good, you're very good, but the drug tails off towards the end of the dose, and you have these off periods through the day, which can be disabling. What you see in the red panel are the actively treated group. Red line is the actively treated group. And over the time course, each 8 weeks, this was assessed of the study, they're getting more and more good quality on time, so that by the end, they're above their baseline and they've got 1.5 more hours of good quality on time. By the point you've reached the vertical line in the center, which was the end of the double-blind study at 9 months, in the blue line, that's the placebo patients, and you can see they're declining. They've got less on time. They're more off, and that was a 1.5 hour difference by the end, which is clinically important, but it wasn't statistically significant. So this could have been by chance. We haven't proven it worked. What you then see in this study is that in the placebo group, at the point of the vertical line, they take the drug, they go to the active treatment, and they then begin to improve and get more on time. And what we think from this type of evidence and other scales we gathered was that 9 months was simply not long enough to have the double-blind study before breaking that. And then that's why in this study, the Regenerate PD, we're going out to 18 months. So that gives us further confidence we may see benefit at this point.
Alan, could you just explain about double-blind?
So double-blind, and this is really important, and I'll come back to the placebo, means that the participant may end up on placebo or may end up on, in this case, the AB1005, which is the GDNF viral vector therapy. So you're randomized to one or the other. And that's something I'll come back to, because that's really something to think about. So you may have neurosurgery and not have the chance of the treatment. Okay, so the study showed encouragement in PET, but lack of benefit in clinical scores. And because this was the 5th trial to do that, although there was encouragement again that we may be on a pathway to overcoming that problem because we can now deliver the drug through the putamen structure, there were questions to be asked, how many times do you crank the handle? But of course, sometimes to make progress, you have to keep going and go from failure to failure without losing encouragement. Now, CP very helpfully put together in 29 a workshop that brought together the key people in the world that work with GDNF. And that happened during this meeting at the Van Andel. And the first question was, should GDNF be tested again? And there was a unanimous view that yes, it should. There was enough here, it was worth going again. But with regard to delivery, how do we get it into the brain? The port was felt to be too difficult. Too often having to come back every 4 weeks, you can get infections around the port. But now you can give GDNF via this gene therapy approach, which, as Matthias said, is single shot, one surgery, and then the virus is continuing to express the gene and you're getting the GDNF continuously. Now, that is something that's good, one-shot surgical approach, but potentially you have to recall that that treatment is then going to keep being expressed throughout your lifetime, and there could be safety concerns around that. So that's something important to consider as well. Now, in terms of pilot work with the GDNF viral vector therapy, there's been 2 studies, and these are what are called open label, which means everybody knows they're on the active treatment. There's no placebo group and they're small. There's safety studies that are done at the outset. And again, just to reiterate what Matthias said, you've got the viral vector, it's given surgically, people are led on the front in the MRI scan, and then the GDNF's injected slowly. And you can see, this is taken from some of those patients, that you're filling that structure. So we know we're getting the drug there, and anyone who comes into this trial will have such imaging during their surgery. Now, the next slide shows the type of participants who were in this study. They're small numbers, and there was a mild cohort of 6 and a moderate cohort of 5 in terms of severity. Now, it would be helpful to tell you what mild and moderate means. Mild means they were very early after diagnosis in a short number of years, 2 years after diagnosis versus 7. And their movement scores, when they didn't have medication on board, which had been stopped overnight, were lower at, you can see the score there of the part 3 at 7.6, the bottom from one row versus 22. Now, 22 is not bad if you've stopped movement overnight. You'd still be able to function with that, but you would be more impaired than 7. So this was this moderate group, but these aren't people that are severely impaired by Parkinson's. That becomes important for when I show you the benefits. Now, next, I'm showing you the safety. Actually, we'll come to the benefits first. Here, we've got the outcome, and what I'm showing you here as the diaries, as I showed you before in the study that was done in Bristol with the port. And what you can see with the diaries is this red line, which is showing you the amount of off time. So off is when the drug's wearing out and you become slow, and then you turn back on when the drugs are working and you can move and so on again. And, oh, I'm sorry.
You're showing the ubiquitous stuff.
I do apologize. Yeah, sorry. What you're seeing here is the amount of off time in the red bar. And what you will note, if you look in the moderate cohort, you've got just over 4 hours of off time. And then after 6 months of therapy, that drops and they've got more green, which is good quality on time, and less dyskinesia, which is yellow. So those moderate stage patients are showing benefit in their diarized scores. So they have better good quality on time through the day. Whereas what you see in the milder cohort is that they're more steady. So they're not getting worse particularly, but they're not improving over baseline. And that, again, is why we've gone for the moderate cohort in the Regenerate PD study, because you've got to be bad enough to see benefit if you're only looking at a relatively small number of participants. You have to look at relatively small numbers in early surgical studies. So we want to give us the best chance of showing this is positive. But if it is positive, then, of course, in clinic, it will be applied not only to moderate stage Parkinson's disease, but also to people with mild Parkinson's disease. going back one, this is the motor UPDRS, and in the red line, so this is med stopped overnight and how you can move. And what you see in the mild cohort is that stays the same. It's not deteriorating, where a higher score is worsening. But in the moderate group, you're actually seeing improvement again, suggestive of regeneration because they're getting better over time. That red line's going down so they can move better. But again, you must remember with this great caveat that this is all open label. So that could be a placebo response. So that doesn't prove anything. But these are potential signals of efficacy. Now, going back to the safety, the good news is that we didn't see, or they didn't see, any serious adverse events, bad side effects due to the study drug, the AB1005. And not only did they not see any serious ones, they didn't see any mild or moderate ones. Now, again, this is small numbers, but for the original safety study, they've gone out to 5 years and it's still looking safe. But when you put the virus in, of course, that's there forever. So whether you run into problems beyond 5 years is not known. So the good safety profile so far, but again, small numbers. And these are things you need to think about at consenting visits. So what I'd like to go to now is the Regenerate Parkinson's study. So this is the latest study. This is an international study with centers in North America, but also centers in Europe, which are in Poland, Germany, and the UK. It's a double-blind study, so-called phase 2, and it's the first double-blind study. So it's the first double-blind study of a drug that is a gene therapy that may regenerate people with Parkinson's disease, where the drug's delivered in this way. So that's what I think makes it very exciting. I think it's one of the key studies that is now open in Parkinson's disease. We'll be looking both at benefits and side effects, and for the reasons I've outlined, it's in moderate stage patients. I have the great privilege, and I do feel very honoured, to be the chief investigator for Europe, and my colleague is at the University of San Francisco, is the chief investigator for North America. Now, this trial shows the trial design. We're looking at 87 patients, which in terms of neurosurgical studies for Parkinson's is large, but of course still smallish numbers, which is why we have to try and optimize as best we can the trial design. And this whole trial design has been riven through with input from people with Parkinson's. So I hope that is, again, reassuring. There's the consenting visit, then screening. If you pass the screening, you then move on to have the one-shot surgical procedure or sham surgery. Now, in the sham surgery, you're anesthetized for the same amount of time, lead on your front in an MRI scanner, but rather than having the virus injected into the brain, you have small, what are called burr holes put in the skull, and it's from the back of the brain, but you don't have the active treatment. So you do have to go through an anesthetic and the skull being drilled into, but they're not going into the brain. So there's some risk, but not the full risk of doing brain surgery. And then those patients who are randomized so that 1/3 will go on placebo and 2/3 will receive AB1005 are then followed up, and they're assessed between 3 to 6 monthly intervals. And at the end of 18 months, it's then unblinded, and there's a comparison between those on the drug and those on placebo to see. And if that then is positive, that becomes a pivotal trial. And this outcome we're looking at 18 months this time, based on the prior study suggesting that 9 months may not have been long enough for enough regeneration to occur to see a difference between the groups. So every step has really been thought about here. And it is a whole pipeline approach to clinic, because it's not just things that have been done for the trial design, but AskBio have been working a lot with regulators, which in the US and the FDA, to say, if this trial is positive, can we move swiftly to phase 3. So to get this treatment to clinic, you'd have to do a bigger study again to replicate the results at phase 3. And the answer is yes, you can move swiftly to that if this is positive. And then if that is positive, the FDA have confirmed that they'll be a way of gazetting this into the clinic thereafter. So it's a whole pipeline approach to get all the way across the river to a treatment in clinic rather than fall over at some point. The active patients who've received the drug will then be followed up after the 18-month point, long term, bearing in mind that the virus will continue, or that you'll continue to have expression of the transgene. But a key thing to know is that the placebo patients may not then, cannot guarantee that they will then get the gene therapy. And the reason for that, we cannot guarantee it at this point, is of course we don't know if the study will be positive or not. We don't know what commercial constraints there will be. So we can't guarantee that there will be a follow-on study so that those people who've had placebo can then have the treatment. And again, this is something to talk to the investigators about should you come to a consenting visit. Now, in terms of eligibility, who are we looking for? So it's people between 4 to 10 years of Parkinson's. So if this is you, you might want to think about it. People who have had their medication, their Sinemet or Madapar stopped overnight and have a score, a rating score of between 33 to 60 on a particular scale. So that's certainly you'd be able to tell they have Parkinson's, but it's not severe. But importantly, you have to have off time. So if you're going to see an improvement in off time, obviously you have to start with some off time. So if you're someone where the drug is, the L-DOPA is keeping you in a good movement state throughout the day consistently, then this isn't for you. But if you're dipping off, so towards the end of dose, you begin to slow and stiffen before you take your next drug. And that amount of time in aggregate, all the minutes that you're off in a day, amounts to 2.5 hours or more, you could be in. You have to respond to the drug L-DOPA, Sinemet or Madapar, to come into the trial. And the reason for that is if you don't respond to L-DOPA, you won't respond to this gene therapy either. And that's because this gene therapy works by potentially sprouting dopamine neurons, and therefore you're having more dopamine in the system. But it, obviously, more if taking tablet forms of dopamine don't help, more dopamine in the system by having your own cells producing more won't help either. And you can't have had brain surgery before. So you've had DBS, you can't come into this. But conversely, if you have this treatment and then later need DBS, you can, of course, have that under standard of clinical care, but brain surgery includes you. So again, it'd be really helpful if you know people who fulfill these type of criteria, you let them know about the study. And then in a study, when you decide if a drug's worked, you've got to have what's called the primary endpoint, which is if you don't make the primary endpoint, the one you've selected up front as your main outcome, the regulator won't approve it. And what we're using is that good quality on time, in part based on the Bristol trial. So that's why prior trials that people take part in, even if they're negative, are very important because each one informs the next step. So it's going to be that improvement in good quality on time to see if there's significant time in the day. And again, the people we're recruiting are those who, when they're on, they're good. They can function well, they can go out to work, they can play golf, but it's when they're off, they're slow and stiff, less able to move. And we want to see that the amount of time they're off and in a good quality on state improves over time. But we're looking at other outcomes as well, which include not only movement outcomes, but also non-motor symptoms, cognition, mood, sleep, and so on, where there is evidence that dopamine in part plays a function in those things. This is just to remind me, this is the European consortium, this is just to remind me that in addition to those scales, we've moved into the modern era, and the intention is to use digital outcomes. So this could include wearables, where you're using something to measure your movement over time in a continuous way, rather than coming up to clinic and having it measured. And a different approach is going to be used in North America. So North America and Europe would use different approaches. It's not completely signed and sealed. We'll do this with the European group yet, but different approaches in those two areas. And that's important, I think, because this will garner information about if the trial positive, this time, which of those digital approaches we should move to with phase 3. So this kind of step forward pathway, so we're most likely to succeed overall getting to clinic, has been thought through. Now this slide, what it shows is the study schedule, and it's really to point out that there are these regular visits at different months, and that there is an imaging burden. So you'll have MRI scans through the study, but also PET scans through the study of different types. So that's a radiation exposure dose. And people can find PET scans uncomfortable, but we're not doing lumbar punctures. On the whole, it's difficult to get people to consent to do lumbar punctures. And we have tried to make the burden as less as possible, but there is a burden there. And people might need to think about how much support from family and friends they have if they want to participate. So in conclusion, wouldn't it be great if we had a neuroregenerative therapy? Wouldn't it be great if we could push Parkinson's back or reverse it? Even if we can't do that, wouldn't it be great if we could hold it so you have movement symptoms that aren't worsening over time? Wouldn't it be great if we could simplify management so people don't need as much medication with all the side effects thereof? And wouldn't it be great if we had a surgical therapy where you could put this into the putamen, but you could still have, for example, exenatide, the diabetes drug that CP have pushed forward, if that's positive, to slow disease down by that mechanism. So in the end, you may have surgical therapies like this, plus disease therapy from repurposed drugs to really try and halt Parkinson's progression applied at the beginning of the condition, but also for others who are more advanced. This drug, AB1005, is a potential neuroregenerative therapy, and it's exciting that this is the first double-blind trial to assess that. And I think it's encouraging that it's a one-stop approach to delivering the drug. So that makes it potentially feasible for nations to give this to people. If you only need one surgery and then it helps forever if it does, you know, that's something that's deliverable. And importantly, we're enrolling people now. So the study's initiated. Sites are already open in the US. By the end of the year or the beginning of next, they should be open in Europe. And I'm going to tell you now about how you can find out more. So if you're interested, or if you could let others know who might be interested, the clinicaltrials.gov website gives more information on the trial, as does the Fox Trial Finder website. And also, perhaps your first point of call would be to email AskBio, where I've given you the email address at the bottom. If you email that, or if you scan in that QR code, that takes you direct to an email. You can write to AskBio, say you want more information, find out how you get referred to be recruited in different centres. In the UK, for example, we're happy for people to self-refer from all over the UK. So you can find out more that way. So that is the end of what I want to say, and thank you for listening. And now Matthias and I are going to answer questions, but Steve Elms and Adrian are both there from AskBio, as well as Matthias, so you can approach them further if you wish to answer questions face to face, or Helen can let you know my email. Thank you very much.
I suspect there are lots of questions. I'm going to come here first. Hang on, do we have a microphone?
Yeah, they're out in the audience.
Oh, very good. Well done.
My question is, after you have the insert, do you still stay on the levodopa? Still stay on your medications?
Yes, certainly initially. So for the intention is for the trial, if possible, to maintain participants at the same level of medication, because you want to look at change over baseline. Now, obviously, if you develop dyskinesia or if you can't manage your symptoms, you may need to increase or decrease, and of course that's permitted. But the intention for the trial is you're stable on meds before you come in, and we try and keep them the same through the trial. The hope would be if people are improved by having their own neurons restored, they won't need as much medication in time. But you have to recall that unlike DBS, so when you switch a DBS box on, you get an improvement straight away because the therapy straight away. But with this, you've got to regenerate nerve cells and regenerate a nerve pathway. And that's going to mean things are growing back because nerve pathways grow back very slowly, which is why we're going out to 18 months. So you'd hope at, 12 months and beyond, and we're seeing those that you'd begin to be able to drop medication. So that's the hope, but not necessarily the expectation. And you have to have reasonable, you have to try and keep equipoise because you don't want the trial to be affected by the placebo effect.
Thank you, Alan. We've got a question at the back.
Yeah, so where are the locations for this? Where do they do the screenings and the MRIs at in the US?
So
And
one
how
of
many
the
locations are there?
Thank you very much. If you go to clinicaltrials.gov, there's a map of the US that comes up with the red dots of where the sites are. Some sites are neurological sites, which will be doing the recruitment and enrollment and the evaluations. Some sites are surgical sites. In some instances, those are co-localized. In the UK, for example, the surgery's in Cardiff, the neurology part's in Bristol, and that's to a certain extent to try and maintain the blind, so we don't run into the surgeon and somehow get unmasked to who had which. But if you go to that, you'll see it for North America. It's not currently on that map for the European sites, but if you email AskBio, they can tell you, and and Matthias and so on can tell you.
We are adding more sites right now. So I'm Adrian, I'm with AskBio. We do have one right here in Michigan. So Michigan State is a local site. So please reach out.
And the search item on that is AB.
If you look for Regenerate PD,
Oh, okay.
that will get you.
Question at the front from Jody.
Hi there. So I have a few questions about GDNF. What does GDNF actually do? So does it actually generate new neurons to replace ones that have died? So if I've lost 50% of my dopaminergic neurons, will it ultimately replace those missing 50%?
Great question. So once a brain cell's dead, it's dead, and you can't resurrect it. But it's to tackle those neurons that are damaged but not yet dead. And it has a number of actions that may cause disease reversal or stabilization, including you get sprouting of the nerve endings, which are the nerve endings release the dopamine, but you may also get restoration of the cell body, which is where the nerve ending projection comes out from. You might also reduce something called oxidative stress, and you might also reduce synuclein, which is the misfolded protein. So there's a number of mechanisms, but we've got evidence from the PET scans and some evidence that I published last year in Movement Disorders that you have got sprouting of the nerve endings and potentially release of dopamine. So those nerve cells are what's called pharmacologically refunctioning.
So it sounds like the best case is I could halt the progression of the disease by sort of stopping further decline of the existing dopaminergic cells, but there's a small chance it might actually improve. Yeah.
The best case is that you'd be improved over baseline. The question is, if you get regeneration, then how much regeneration will you get? Now, by the time you present, of course, what we know from PET scans is that you've lost 50% of the dopamine brain cells. And what we do know from the PET scans is that people's improvement progresses over time. So as the months go by, you see the PET scans getting better and better. That's been shown in earlier GDNF studies. But what we don't know is if that keeps going or you reach a certain point. But the best case scenario would be a degree of disease reversal. Second, and then holding at that stage. But that's best case scenario. And you have to have equipoise. But obviously, if you held where you are, that would be good, particularly if you applied it early in disease. But as I say, at this stage, we're looking for those patients who are functioning but have some off time.
Great, thank you.
Jody, what GDNF takes advantage of is the fact that during the pathogenesis of the disease, it's not like a direct move from on to off, from cell alive to cell dead. There is a large gray zone in between. The cells go, if you wish, into hibernation. They start to sleep and they start to lose their normal function, their phenotype. And those cells, which we call sick but not dead, can be resurrected by GDNF. So GDNF helps them to regain power, to regrow their axons, to initiate their normal function in terms of dopamine production and everything else. And that, how big that population of cells actually is, we don't know. That's difficult to assess, but it's certainly a sizable population, assumedly in this case. And that's the target population for the GDNF therapy.
We have questions. Thank you.
Yeah.
Thank you for that. These might be simple questions. How do you ensure that the vector doesn't go into a wrong cell? And how do you guard against mutation over many years that could lead to some other form of disease like, I don't know, cancer or something?
It's just the nature of certain viruses, in this case, the AAV2 viral vector, to attack certain target cells. So the AAV2 vector is known to specifically attack brain cells or nerve cells rather than any other cells. Or to target other cells, you would use other AAV0 subtypes or even other viruses. So we know for a fact that AAV2 preferentially clicks onto nerve cells and releases its genetic content into these cells. Now, as I mentioned during my talk, the transgene in the case of AAV is known to be stored in separate vesicles within the cell body rather than within the nucleus, within the cell's genome. And in order for gene therapy to produce cancer, to become oncogenic, it actually needs to integrate into the genome so that the cell, when it divides can deliver the genetic information to the next generation. Another factor that protects from that is the fact that neurons don't divide anymore. So you have two protection shields against oncogenesis in that case.
Thank you.
Another question at the back.
Hi, the primary endpoint is the PD motor diary, correct? Okay.
Yes.
Is that a patient-reported outcome, and how frequently is it collected, and is it a novel endpoint, a primary endpoint?
No, so the diaries themselves have been an outcome that's been used for many years. The diary is divided into half-hour epochs, and the choice is I'm off, I'm on with mild dyskinesia, which is a good quality on, or I've got problematic dyskinesia, and the participant has to tick each of those for half an hour epochs, and they're supposed to do it in real time, and that's collected over the 24-hour period. And the reason for using that as the primary outcome is that was the strongest, one of the strongest signals in the phase 1b study that I was showing you. It was also the strongest signal in the port GDNF study, and it's a patient-related outcome, and it has a real-world utility. Whereas if you looked at the motor UPDRS, you stop your Cinemet and Madapart overnight, you see how poorly you can move after not taking your pills at all for 12 hours. Now that might tell you if you've got disease improvement or reversal over baseline, but that's not something you're going to do in reality. So it's not a real world functional improvement. Very importantly, the regulator has to decide whether this is meaningful to give approval for the drug, and the regulators in the European and US both all said we want to see this improvement in good quality on time. They've moved away from other things. Interestingly, in the port study, when we composited together and looked at good quality on time, and this is with Matthias's work, and added in what happened in the not taking your pills off overnight, when we added those various components together, the study became positive, but that's not a validated scale. So there are signals that this may work, but we don't know that it will work. We have to keep equipoise to avoid the placebo effect, which could scupper things. But that's how we arrived at this outcome. But in addition, as I mentioned, in an exploratory way, there's going to be these digital things, which of course record for longer periods of time. If it's a wearable device in Europe, we might use a wearable in the lumbar region of the back. So that gives more continuous data. So the more times you sample, the richer source of data you have, but the regulators aren't yet ready, set up to say that yes, an outcome in a digital will be enough to go forward, but the diaries are. Does that
diary at home? Yeah.
Following up on Alan's last point, the motor diary relative to the UPDRS motor score has the advantage that it looks at a small longitudinal period. As Alan mentioned, it has to be the on-off times have to be documented in half-hour intervals over a 24-hour period. In fact, over 3 days altogether, and we're looking at the average total amounts of off time per day in over that 3-day period. So this is a little bit of a longitudinal aspect, whereas the motor score is strictly cross-sectional at one particular time point. And that is subject to a lot of variability depending on what you've been doing the day before, how your night was, how you're in the morning. So there is a lot of variables that have impact on that, and the diary has its own pros and cons, but this certainly is a significant pro.
Yeah, and adding to that point, all of which was very helpful. The motor score, the UPDRS, is an in-clinic. So you come up to hospital having stopped your meds, you might be videoed when you do the assessment and so on. But that's, as you say, at one point in time, but it's in an artificial setting of being in the clinic. The diary's at home. But it is the case that some people can't really tell when they're on and off. And some people aren't very good at complying with assessments. And so those are part of the selection criteria. So there'll be training if you come into the study to use the diary. And there's a step in it where for a period you'll be it'll be assessed between the person during a period of time when they're in clinic and the patient or participant is rating in the same way as the clinician. If you're good on that, then you'll be enrolled into the study.
Question back.
Marie.
Hello, I have two very different questions. The first one is, how did you choose the number of viral particles that you're going to inject? And because obviously you're going to have a permanent stimulation, permanent release of GDNF. And aren't you afraid that with long times you might exhaust cells by always telling them to sprout? And that's the first question. And the second one is about women. I imagine that you want a 50-50% recruitment in your trial. How are you going to control for hormonal influence in women? Is that a confounding factor that you're taking into account? Thank you.
As relates to your first question, as Alan mentioned, there were, well, number one, there were a number of preclinical studies. So before the first person with Parkinson's was even exposed to AAV2 GDNF, a whole number of animals were treated. And we had initial information that guided us in terms of how much we should be delivering to people with Parkinson's. And then, as Alan mentioned, there were 2 preceding phase 1 open label studies, one with 13 patients and one with 11 patients. Alan has been focusing predominantly on the latter study, including 11 patients, but there was a previous one that had 3 dosing cohorts with different dose levels of the virus. And we initiated the second phase 1 study only after we had readout from the first study over several years. So there was significant information available to us that helped us determine which is the right dose level to use in the previous study. And in fact, it turned out that this dose selection was sort of right on the money, because the dose we used in the phase 1, in the second phase 1 study, is exactly the same dose that we'll be using in the Regenerate PD study. In terms of your second question, a short answer would be we take what we get. So we're not requiring a certain split between males and females. We're enrolling people with Parkinson's as they come, and according to the natural split within the population of patients, of people with Parkinson's. And we're not specifically controlling for any hormonal influence on the treatment outcome.
And I think this probably should be our last question, because I'm sure everybody needs a bit of a warm up, actually.
Sure. So, yeah, real, I think it's easy. How many people do you anticipate being in the study? When does this study start? Will you be starting patients in the routine, in the exam and study itself while you keep recruiting, or do you wait till? So I'm curious how big this study is worldwide, worldwide. And then you maybe can't answer this, but how is what you're talking to us about today going to, what should we think about when we hear Roger Barker speak in 15 minutes about stem cell? And I've heard him talk a little bit before, so I know he goes through the history of all of this, but so maybe that's a difficult question, but maybe you could just say how what you're talking about relates to what Roger is going to be talking about. Maybe he emphasizes more the stem cell history and that kind of thing.
Alan, can I start with that, if possible? So the very first slide that Alan showed was actually a good questions to ask guide that has been developed by the University of Cardiff, which was very much to enable people to ask really good questions about the studies that they've been writing down. And I think that probably the thing we need to do is to circulate that to everybody who's here today, so that you've got a chance to actually look at that and see whether there are other questions we ought to be adding to it, actually, as well as anything else. But I think you have to look at the studies in their own right as to whether they are right for you, and ask those questions about the studies themselves for the investigators who might be recruiting you. Because I think it has to be very much about information shared, but also the opportunity to ask and answer questions so that you're identifying, the things that you want to know about, but also the risks and benefits that you might see coming through this. And benefits around clinical research. I touched on this yesterday. In the UK, it's actually the chance to see a neurologist rather more often than you might be able to normally, for example, and to ask questions about your own disease. That's one of the known benefits of taking part in research. Whereas I think some of the risks are in surgery. Both these two approaches are surgical approaches. So those are the sorts of things you need to be thinking about when considering these trials. They are not. You need to go into them with your eyes very clearly open and with lots of really good questions to ask of those investigators. The other thing I would add to that is that, we talked a lot about communication yesterday and the importance of communication. And given that recruitment will not be done and dusted in a day, because it's a surgical trial, making sure there is excellent communication with participants is going to be very important to keep everybody involved and engaged. And so thinking that through AskBio, please, would be wonderful. That's a special plea.
Yeah, thanks, Helen. If I could answer the second question first, and to answer it, perhaps to tell you what the similarities are. So all neuroregenerative therapies require a surgeon at the moment to get them into the brain, apart from some early work with ultrasound to help get things in. And they break down into, as you've said, cells or growth proteins. And at the moment, as far as I know, this is the only phase 2 growth protein study that's going. So it's cells or growth proteins, and both of them in the same way are forever therapies. So once you put cells in someone's brain, or you put a virus in someone's brain expressing a transgene, or a transgene is in the brain, they're both there forever. And both of those approaches have been not shown proven benefit at several studies. In the cell ones, we've only got the fetal graft studies that have been at phase 2 so far. But in fetal graft studies and in the protein studies, they've not shown. But in each case, each study has led to new information. So it's like bricks in a wall. And both now, because in the cell therapies, you're now using induced pluripotent stem cells, are using patients, are growing, if you like, patients' own tissue. The only difference that I can think of that would, well, there are a number of, but one potential benefit, or two potential benefits of this approach over cells is that there may be a benefit in regenerating your own neurons and the way they reconnect than implanting neurons. If you just dump cell bodies into the putamen, you don't know they're going to connect back up in as well and restore pathways as if you restore or regenerate your own endogenous neurons. And you could ask Roger about that. And the other is that for some of the cell therapies, you have to damp down the immune system because you've got foreign tissue, where those drugs that damp down the immune system have some risks. So that's that one. I don't know, Matthias, do you want to take the other one about how many people and so on?
Yeah, one further comment on what Alan just said. I think we're just in a luxury situation at the moment, in a sense, as we have two different regenerative approaches, although the real regenerative approach is gene therapy, while stem cell therapy is not regenerating your own system, as Alan pointed out. But in rough terms, those are regenerative therapies, and we're just very lucky that based on phase 1 studies that came out with pretty comparable data, we're now pursuing two different approaches that really come with the hope of changing the treatment paradigm for Parkinson's disease. That is really something that makes me very optimistic, and I just hope and expect that one of the two at least will make it to the finish line. In terms of your other question, we're aiming to enroll a total of 87 participants, collaborators in our study across the 4 countries mentioned: US, UK, Poland, and Germany. We have a few more sites in North America than we have in Europe, but the final split is not determined in terms of the 2 continents. So we'll have to see how recruitment works out. In any case, recruitment will be ongoing for probably about a year and a half or 2 years, depending on how active the Parkinson's community actually is in terms of volunteering and being courageous enough to enroll in the study.